the major key points here is actually why the biosimilar is looked for a cost-effective alternative, if you all agree with that. But otherwise, there is no need for that. So the main issue comes on the cost. Otherwise, we all know that original molecule is always superior. There is no doubt about it. So let us start with this, what Ashish has given me the information earlier. So they have always, all the development and validation if you go through, they have the expensive preclinical, clinical trial and extensive, not the expensive also, but the extensive clinical trials, which is compared to the biosimilar trials, is a much more few studies, smaller sample size, and their majority of them are retrospective analysis. So here lies the challenges of proving the equivalence of the safety and efficacy. So they have manufacturing process. If you now go to the basic of that. So if you go to the manufacturing process, the original molecule they develop from the original cell line. And they have only one original cell line they keep using for every batches of that original cell line. Now what happens to the biosimilar? There is a variability due to the reverse engineering process, and they have a different cell line they use, and that is the reason they develop the immunogenicity issue, the toxicity, the endotoxin, everything comes from there. So there is a basic difference between the manufacturing process. You all need to understand that first. Number one. Number two, if there is a variability in the manufacturing process, the biosimilar, go to the term biosimilar, it is biologically similar. It is not the same product. I repeat, it is biologically similar. So there has to be a difference. What are the differences? You need to know first, then only you will understand it. So these are the few points I will just describe. Biosimilars are a large molecule compared with the generics, and mentioned previously, they are not based on the fixed clinical formula. Originator biologics do not provide the complete information in terms of process followed during the manufacturing even after expiry of their patent. So that means even after expiry of the patent, they are not bound to give that exact process of manufacturing. And that's why the biosimilar makers, they do not get the exact manufacturing process. They just try to imitate them, and how they get the, once this patent is over, they get the molecule, and by reverse engineering, they do it with the different cell line. That leads to different immunogenicity, and that is in the initial hitches we do see in every cases of biosimilar. And the immunogenicity can be an issue. Note this point, immunogenicity can be an issue with biosimilars due to different living cells and the process followed by the different biosimilar developer. And who has given me this data? Dr. Ashish Sharma. This is, I have quoted from his publication only. Now this is, we all know that we have the extensive data for every innovator drug up to 5,000 of the patient level. So which cannot match to any biosimilar, I think so. So you have a extensive, robust data. You all have heard Marina, Anchor, Pyre, Resolve, Restore, Reveal, Refine, Bravo, Cruise. I think each and every who does the practice of the medical retina, they know it. They know it very well. But how many of them do you know about the biosimilar studies? Do you actually? So anyway, I mean, and you have the rise and ride, like these kind of trial, they have the horizon is even more. So they have the 24 months of the safety data available. So we don't have that kind of, over the horizon, the safety data available for the biosimilar. So the issue comes with the safety and the cost and efficacy. These are the three words I will talk about. The regulatory system is much more rigor for these things. Even FDA and DMA, they mandate for the complete molecular characterization for the clinical studies. Whereas in biosimilar, they have only two steps. One for the FDA approval, that is analytical data, and just do a phase 3 trial and get out of it. So this is very, very easy to get a biosimilar to the process, whereas the original molecule, they go through a rigorous data. These are the information why I am giving to you, for you to understand that how original molecule they do develop and how much of hard work, and the safety protocol is so stringent for them. So as usual, what I have described, there is a batch consistency for original molecule. There is definitely there is difference between those two from batches to batches. That's how. So if there is no cell line, fixed cell line, so if you keep on changing the cell line, the toxin level and the toxin molecule, every time it keeps on changing. Depending on that, the immunogenicity becomes also an issue, and it causes a variable immunogenicity in different phases. So you know that this batch is safer, but next batch you don't know whether that will have the different immunogenicity or not. So in a clinical, in a PAT study, a survey study, it has been seen that 78% of the user of those, medical retina user, they preferred for the original molecule rather than the biosimilar because of the safety and the medical legal issue. Even they know that this is a cost-effective option. So this is how the treater minds, I am just reading for them. So even they have the much more, and the revenue, yes, for this revenue, yes, there is a little amount of the more revenue is involved into there, and that revenue again goes to development of the new drug or the new formulation of the drug. That gives us another boost for the going to the next generation. So there is always a proven history for over the decades, more than 15 years of the original molecules. They have this for this Ranibizumab, and which we are using safely over a period of time. I never had, and all of you I think vouch for that, we will never have regarding the safety issue. So whenever you prescribe a drug of the original molecule, it comes to your mind about the cost, not the safety. Whenever you prescribe a biosimilar, the safety issue comes to your mind first. If everyone is here, you put your hand on your chest and then accept it. So to summary in nutshell, so if you have what Ashish was telling, that yes, for my mother I will use, but if you have a 20% of the more amount of the money, if you can involve for more amount of the safety, I think all of you, for your safety, you do term insurance, blah, 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 blah. For 20% if you increase, the safety becomes much more secure. That's what you just use it, and I will prefer for that. So in nutshell, ultimately,
you can sing.
और जो बेटा बाप से पैदा होता है वो बेटा ही रहता है.
Very well. I know it was a very difficult job to, you can sit down.
Sir, I want to ask one question to the audience. So how many of you are preferring originator rather than biosimilar? Raise your hand.
Just show of hands. We want how many innovator and how many biosimilar?
Or biosimilar?
So Ashish, you have 1 minute
almost
rebuttal. You can go there, because the reason I wanted 1 minute rebuttal was you can take a mic. Take mic. You see, today Rupak Kanti, even for the purpose of debate, is still bothered, and he quoted your publication also. He is bothered for consistency, he is bothered for batch variability, and he is bothered for immunogenicity. So your comment on this.
So I think it's a very important point. So let me just clarify one by one. First that Rupak said, they use different cell lines. Absolutely wrong. Same E. coli is being used by Innovator, and the same E. coli is being used by the biosimilar as well. And the point which was quoted that you use every time different cell line, again wrong. One cell line is used, and that is preserved at the 5 locations, not only in India, across the globe, in America, in Europe, everywhere. So that if some fire incidents happen, something happens, we don't lose that cell line. So these are important points, that's where we need to clarify. Second, batch to batch variability. Do you remember the slide I showed of Lucentis? It started with 18% inflammation. What happens now? No. Not even Lucentis, Avastin, Brolucizumab, Faricimab, no, you cannot have each batch same. There is always a variability in each and every batch. Whether you talk about Lucentis, each batch, you can check it. Because biologists cannot produce the exact copy. So that is second point. Third point, phase 3 trial, truncated phase 3 trial. So I mentioned, this was our conversation with the FDA reviewer, Wiley Chamber. when it was launching in Europe, that Ashish, you need to clarify to the US population that why these phase 3 trials are truncated. Because as a retina specialist, we are used to see big data. And if you again try to do big data for biosimilars, the purpose of biosimilars is defeated. And you are trying to make the similarity of a molecule where the efficacy, safety is already established by Innovator, so there is no risk there. And another point, which sometimes people have negative perception, only one indication, the trial is done. Why? If Lucentis would have shown that RVO, DME, AMD behaves differently in safety, regulatory would have asked another trial from a biosimilar that you do the trial, but they don't do. So extrapolation happens.
Okay,
so all of us, we need to understand this.
Last 10 seconds, anything?
Yeah, so we need to understand that. That's why the second last slide was education, and understanding. Once you have that, I think that's the way to go to serve our population.
Rupak, can you please stand and tell honestly now, you still treat majority of patients with innovator or biosimilar? Honestly.
I will, if I can show my prescription there, I give the options of, I'm not sure how many of them, I always give options. There are few, they are laughing, they know it. So I give the options of innovator oblique biosimilar.
You see,
And
one second,
70%
my argument here is, you see, patients in this country will ask you, Dr. Sahab, aap batao. You see, you tell patient, this is a matter of confusing the patient. Aap ye le lo ya ye le lo. So that's not correct.
Sir,
what do you do?
Sir, if you ask
If
me,
it is your nana or mama.
I will go for the original molecule only.
Okay. So we'll have one comment. One comment from Gopal. Only 1 minute.
Yeah, sir.
Innovator, biosimilar.
Practically in my practice, now I have almost given up innovators. I am into biosimilars. No. But you have to differentiate between science versus the business of treating blindness. Not just for the ophthalmologists, but also for the health administrators. So that.
But in any case, you trust biosimilars.
It depends on the company. It depends upon the reputation of the company. It depends upon the quality that they have shown.
Since morning you are giving political answers.
Given a choice, if the patient affordability is not an issue, obviously innovator.
Ask Abhijit.
Question, sir, means biosimilars.
AG, you can start from there. You see, in our analysis also, you see the usage of innovator has dropped down to less than 10%. And biosimilars have risen as number one molecule. Not because, today, believe me, Rupak, there is no fear at all. Depending on the company and all, you can, have, like Rani Eyes had an issue. But believe me, that was a procedural issue, not a because of the molecule. So initially, aflibercept also, as he was saying, 18% inflammation used to occur. And since the time that ISO limit has dropped down, there's hardly even Razumab, 15 we had a couple of, after that not even a single patient.
Yes, sir.
So today, if you go by what you were saying that you give an option, today I do not give an option. I do not give an option. We say this works because there's a made in India pride also, which this works as good, believe me, as good. If you tell a patient, ये भी अच्छा है, ये imported है, ये Indian है, वो तो फिर वो कहता है मैं imported ले लूँगा. But generally speaking, 0.00014 के लिए we do not, because 60% cost reduction has to be there, honest cost reduction. 4,000 versus whatever 12,000 he said, that figure has been quoted in American Academy also. One last comment, politics.
Yes, sir. As far as efficacy is concerned, head-on-head phase 3 trials, good efficacy is shown. But these studies are not powered for safety. And retrospective reporting of complications are much lesser than prospective studies regarding safety. That's the only point I want to make.
Last comment by our chairperson, then we'll go for the third debate.
Now, obviously, first line of treatment should be by the innovator, and you should always see the probability of these biosimilars, the potential of these biosimilars in the coming future.
No, because if India started this story, the whole world is now accepting it. USA has approved it. If you were talking of stringent criteria, believe me,
But
there can't be more stringent than US.
Time-tested
Even in ROP child has to live for another 80 years. So there also. So in any case, we will not go into debate more. The answer is biosimilars have actually now ruling the market now. There's no doubt about it. It's not only money, it's equal efficacy and equal safety. In any case, we'll go on. Rest we can talk over later on.